Hundreds of thousands of researchers around the world are working to improve life and address imminent threats to humanity. Often their research ends up in the scientific valley of death in the form of publications and patents that never see the light of day. That is about to change. Welcome to the Lab to Startup podcast, hosted by Naresh Sunkara, founder and executive director of the Berkeley Postdoc Entrepreneurship Program at the University of California, Berkeley. This show has two main goals: share the stories of those who have successfully founded startups based on their own research, and highlight resources needed to help those aspiring to launch startups in the deep tech space. Whether it's electric cars, vaccines, addressing climate change concerns, or possibly establishing life on other planets, Naresh and his esteemed guests want to help scientists, engineers, faculty, and researchers bring their innovations to market. Learn more and subscribe today at labtostartup.com. And now, here's Naresh.
My guest today is Irene Griswold-Prenner, the founder and chief scientific officer of Nitrase Therapeutics. Nitrase is a biopharmaceutical company unlocking the therapeutical potentials of nitrases, a new class of enzymes discovered in-house. Their platform offers a promise of pipeline of therapies against a broad variety of diseases, starting with Parkinson's disease. Nitrase Therapeutics has been widely recognized and has won multiple awards, including the prestigious Target Advancement Grant from the Michael J. Fox Foundation for Parkinson's Research. Scientists pride themselves on being keen observers, which prompts curiosity to identify a problem and persevere through challenges to find a solution. Irene is the true definition of a scientist who observed a biological process called nitration of proteins about 20 years ago, but no one knew how the nitration was happening. A number of people dismissed it as another chemical reaction that happens in the body. She knew that post-translational modifications of proteins play key roles in the regulation of biological processes that lead to various physiological responses. Her scientific training made her dig deeper to find out how nitration was happening. All she had was an idea and hypothesis that if she can find the cause of nitration and how it affects different metabolic activities, she could potentially develop therapeutics for the underlying conditions. In this episode of Lab to Startup, we will learn about Irene's scientific training, her professional experiences working for other pharmaceutical companies and startups, to get to the bottom of nitration by launching Nitrase Therapeutics. Irene's story is super fascinating, especially the way she was able to pitch the idea in its true sense at that time, raise some seed money from non-biotech investors, and find a new class of enzyme called nitrases, which led to raising about $45 million in Series A that could potentially help develop new therapeutics. I'm really excited to learn and share from Irene's experiences. Hope you enjoyed this conversation. Irene, I'm super excited to have you on the show. You have a very interesting career path till you launched the startup Nitrase Therapeutics. And since your past career and the current startup are interconnected, we might go back and forth to connect the dots for the audience here. So let's start with what you're doing at Nitrase Therapeutics. Why the name and things about NITROME platform?
I would be glad to. My name is Irene Griswold-Prenner. I'm currently the founder and CSO of Nitrase Therapeutics. We were fortunate enough to bring in a CEO to take over that role from me. His name is Pierre Beauring. We formerly were called NITROME Biosciences, and part of his evaluation as he came in was we changed the name from NITROME, which is the representative of the class of nitrases, and we changed it from NITROME Biosciences to Nitrase Therapeutics to signify that we're focused on developing drugs to benefit patients that are focused on inhibiting nitrases. That's the reason for the name. And so let me just tell you a little bit about nitrases, the NITROME, and how we came upon this. The nitrases are enzymes that catalyze a post-translational modification called nitration. It's an NO2 group that's added onto tyrosines. So just as phosphorylation kinases can put a phosphate group onto their substrates and change anything from structure to function to activity of that substrate. The same is true for nitration of tyrosine residues on proteins. They can change structure, function, or localization of the protein. This was previously thought to be a chemical reaction The cofactor in the nitration event is a free radical called peroxynitrite, or an oxidative stress product called peroxynitrite, and it's labile. The idea previously was that nitration was just a chemical reaction as peroxynitrite is decomposing. If a tyrosine happens to be nearby, a nitrate group is added on. I realized in some research I did a long time ago that there was likely an enzyme that was involved in this process, and Nitrase Therapeutics was started with the idea of identifying this class of enzymes and seeing if we can drug them for therapeutic benefit.
Very interesting story. Are there specific disease areas that you're trying to target at the company?
I started out with the idea that there was enzymatically driven nitration of alpha-synuclein. Alpha-synuclein is a protein. There's mutations in alpha-synuclein that cause Parkinson's disease. It's also increased in levels. It accumulates in Parkinson's disease. So alpha-synuclein and Parkinson's disease are our first therapeutic target. But we're now developing into oncology, possibly lung injury, as well as autoimmune indications are a possibility as well.
What was so interesting or unique about this nitration mechanism that caught your attention?
The way I got into this was back in the Elan days. So I first came up with this idea 18 years ago. And what I was researching at the time was trying to understand what the signaling pathways or the molecular mechanisms were that contribute to Parkinson's disease and models of Parkinson's disease. And what I realized in researching was that a big component of Parkinson's disease was obviously the aging component. And one of the big things that happens in the aging process is an increase in our body's levels of free radicals, so both oxidative and nitrative stress. And in research, I realized that this had to play some major role in Parkinson's disease, not just synuclein. So when I started to study this aspect of it, what I realized is the main end product of oxidative and nitrative stress is nitration. With that being the case, I wanted to understand what nitration might do in Parkinson's disease, and started reading the literature and found that there were, in both models of Parkinson's disease as well as the disease itself, there are really only a couple of proteins that were identified as having any level of post-translational modification of nitration. And those two proteins at that time were alpha-synuclein and tyrosine hydroxylase. Both of those proteins are very important in Parkinson's disease. So number one, it was interesting that a post-translational modification was occurring on proteins that are important in Parkinson's disease. But also, if this was just a random chemical reaction, then it wouldn't make sense that we always had just a couple of proteins showing up that are actually important in the disease. What it sounded like is that there was a directionality to what proteins are actually being nitrated. And usually when that's the case, it's an enzyme that's actually catalyzing this event. And as I researched further, I learned that nitration actually decreases tyrosine hydroxylase activity by 50%. Which would be problematic for Parkinson's. And nitration of alpha-synuclein causes it to aggregate and kill the neurons. So this says that if there truly is an enzyme that catalyzes these events, then by inhibiting it, you could actually have a disease-modifying therapeutic for Parkinson's disease. So at that point in time, it was just a hypothesis, but it was a hypothesis that made sense, and there was a potential for therapeutic indications out of this. So that was the reason I got interested in it and did some early work that was suggesting that there truly was an activity that catalyzes nitration of alpha-synuclein.
Interesting. You mentioned Elan Pharmaceuticals and you mentioned a long time ago. Let's put things in perspective for the audience. When was this and what were you doing at Elan?
When I first came up with the idea was in 2002. So I was just probably still a scientist, maybe senior scientist at that point in time. I came from an oncology background, signal transduction background, but I was brought into Athena and then Elan to understand the signaling pathways that go haywire in neurodegenerative disease. And my lab had already studied that for Alzheimer's disease, and then we were asked to do the same thing for Parkinson's disease. And this was the puzzle that I kept on coming up with Alzheimer's. Amyloid beta induces a neurotoxic signaling pathway. So there's a series of proteins that are involved in the signaling pathway that lead to death of the neurons. It wasn't so obvious in Parkinson's disease. What was obvious is that oxidative stress, free radicals, are an early problematic feature of Parkinson's disease. So how do you tie that into what's actually occurring from a molecular mechanism in disease? So that's when I came upon this idea. And the good thing about the time at Elan is that we had our goal, as with any pharmaceutical or biotech company, was to develop drugs, but there was also some flexibility in studying things that weren't completely on critical paths. So I was allowed to have a pilot effort where I had people come in and help me every once in a while to see if this was actually a possibility. And we got enough data at that point in time. So between 2002 and 2005, on the side of doing every other project we were working on, to think that this was actually a possibility. Not enough to actually advance at that point in time, but certainly enough to think it was a possibility.
I'm so glad you had the flexibility to do a side project. Otherwise, this would not have happened. This is very early in terms of Parkinson's. 2002 is almost like 20 years ago. And you came across that nitration affects tyrosine hydrolases. How widely known was that information, and what was anybody else doing in this space?
It was actually nitration, peroxynitrite levels in disease and aging, and nitration was actually studied quite a bit in the 1990s. And actually one of the initiators of this field, Dr. Harry Istropoulos out of UPenn, is actually now scientific advisor of ours. So he and a number of other colleagues had discovered that peroxynitrite is a true endogenous factor. It's an end product of oxidative nitrative stress in the human body. And they realized that nitration was a major endpoint from peroxynitrite. So they and others had studied nitration of alpha-synuclein and shown that it drives aggregation and death of the neurons. And then other groups have looked at nitration in other indications on other proteins. But the field sort of just stayed static, or I'd say even fizzled, because if this was truly just a chemical reaction, what do you do with it? It was my idea in 2002 that there was an enzyme that was involved, but I wasn't able to get back around to this until 2017 when I started fundraising for nitrases. If you look at even review articles on nitration that are published, there will be just a blanket statement, one sentence in each of the reviews, that protein nitration is a chemical reaction. Period. That's all that's ever discussed about it. There's some papers that talk about the fact that you have a particular pKa of a tyrosine that's more prone to nitration, and that nitration is often on the same tyrosine residues that are tyrosine phosphorylated. But still no one made that connection to the fact that it might be an enzyme that was actually catalyzing that event because of the chemical reactivity of peroxynitrite. No one thought that an enzyme could beat out the decomposition of peroxynitrite. I think I was just naive enough to think that that was possible.
Naive you were. That is a fascinating story. And what happened? You realized that there could be an enzyme actually causing this nitration. Who did you first talk to? Did you talk to anybody at Elan, or what was their response?
I did. I talked to the heads of research at that point in time, and honestly, they were quite supportive. I was able to bring in a postdoc to help me out with this for a period of time. There were definitely the naysayers. There were those who just thought there was no way this could be possible and thought it was silly that I was even trying to go after this. And even when I showed them what I thought was actually pretty compelling data, not identification of the enzyme, but showing that there was a protein activity that increased nitration and aggregation of alpha-synuclein, it was still just a blanket statement that no, this can't be the case because that's not what the field says is going on. So there were people who were very supportive, and there were people who completely discounted that there could be a class of enzymes, even at that point in time, so almost 20 years ago, that no one else had actually thought of or characterized, let alone now. During the Series A round of discussions, one of the main questions I got was, how could we have identified a class of enzymes that no one else has discovered or even thought about, for that matter? But I think you just have to be asking the right question at the right time.
The right question at the right time. You bring up an interesting point about the naysayers. We entrepreneurs face naysayers all the time. How did you, I'll come back to this subject again, but how did you cope? Because we believe what we want to believe, we humans. We tend to be wrong sometimes, a lot of times. But then you're getting both sides. And how did you keep that balance? And how do you keep something like that alive, an idea from 2002 to 2017 or whenever you started this company? What was the coping mechanism?
I've developed a thick skin over the years. I think you have to have, as a scientist, you have to be able to listen to input and critique. I think you have to be your biggest critic to make sure that whatever you're working on, you haven't forgot a control or you haven't missed something that you should have picked up on. But at the same time, if you truly know that this is something that's going on, I think you just have to persevere. And for me, even before I had data on this, I knew that there was this class of enzymes existed. I just knew it was the case. And I knew it was something that no one else was going to figure out, and no one else was going to put the time into trying to identify this class of enzymes. There was nothing easy about this process. I think if I didn't have a thick skin to begin with, it probably wouldn't have been able to. But that, together with the fact that I knew this class of enzymes existed, made it so that I thought about, I call them nitration enzymes at that point in time. I probably thought about them every day from 2002 out to, well, I was going to say 2017, but out to today.
That's a great character. You bring up a few points. Definitely have to grow some thick skin as scientists and entrepreneurs, because you're going to receive those no's all the time. And it is important to listen to the inputs as you talk to people, both the good and the bad, and be your own critic for sure. Be truthful to yourself. I think when you have an idea, when you cannot sleep for such a long time or that, I think you should definitely pursue that. And I think startup is the best way to bring those ideas to life. Thank you for that. We'll come back to those naysayers again. So what happened after Elan? I know that they got merged with Perrigo. What was your career afterwards?
Even before that happened, so Elan started to have financial difficulties for a number of years. From the time at Elan, I moved over to a company called Hyperion. I was intrigued. I'm really not a big technology person, just for the technology's sake. This was when iPSC, or induced pluripotent stem cells, had just been discovered by Yamanaka, or shown how to make them from human cells. And it was just starting to be shown that you could actually see disease phenotypes manifest in culture. And to me, that was phenomenal, because you're not, with any type of mouse model, you're biasing your cells by expressing a transgene. You're assuming what the disease is. What I liked about the idea of the iPSC cells is that you're actually watching, at least if it's a de novo phenotype, a phenotype that's manifesting based on what the patient cells are telling the cells to express, the changes that occur. So I thought there was a lot of potential with that technology. So I went to Hyperion. I was there for, I think, 4 or 5 years. Got to the point where I ended up leading an effort in which we had developed an Alzheimer's model of disease. We'd found that there was a secreted form, specific type of secreted form of tau. We made an antibody, function blocking antibody against tau, and we ended up having the first tau antibody into clinical trials, as well as the first therapeutic from iPSC approach into clinical trials within the 4.5 years that I was at Hyperion. It was a really rewarding experience to be able to see program, a therapeutic development so quickly in a very new technology. And it was just an amazing group of scientists that I was working with there. So it was a really great experience to work at Hyperion. But then we were bought by Bristol Myers Squibb, and my husband and I decided it was time to take some time off. We'd both been working very hard for way too long, and thought that we would take time off until we could figure out what to do next. In the meantime, ever since I'd left Elan, there was a program that I had run at Elan that I was trying to get out of Elan. They had to shelve a lot of projects as their funding decreased. And there was a project I just thought didn't make sense to be sitting on a shelf. So I, every few months, contacted the head of research and head of legal and asked them if they were ready to out-license the program that I had led there. And they were never able to. They always wanted to be able to make sure that they could turn everything around. But it wasn't until, I think it was just a few months before Hyperion was sold to BMS, that Elan was sold to Perrigo. And Perrigo is a generics company, so they had no ability to take preclinical programs forward. As my husband and I were going to take time off, driving to Jackson Hole, Wyoming to relax, that my partners and I, Karen Chen and Diana DeVore, finished the discussions with Perrigo that allowed us to acquire all of the former Elan's preclinical asset and to be able to advance those. So that included the program that I was interested in, the J&K program, but also all the other preclinical programs that were remaining at Elan. And this was the summation of years of, I should say, small molecule preclinical programs. The rest was at Prothena. And this was years of work, really nice scientific and drug discovery work that would have sat in the shelf otherwise. So I was like, well, I could take time off, or this is the one time when we can actually take forward the program that I want to. And we signed on the dotted line, and that was the initiation of Imago Pharmaceuticals. Unfortunately, probably actually fortunately. I was just then, had just moved to Jackson Hole. Karen Chen was in New York City and still full-time CEO of the SMA Foundation, and Diana DeVore was in Denver. The programs were sufficiently advanced that we thought that we could run the company virtually and advance the programs that we chose to do so with CROs. And what we did actually, we also stayed completely non-diluted. We took in no investor funding. We funded the company through grants and primarily through selling the programs that we couldn't take forward ourselves. And selling those programs, grants, was actually what funded the development of the J&K program that I was interested in advancing. We had started Imago with the idea that we would run ourselves out of business at some point in time. What we didn't want to have is these programs sit on a shelf and not go anywhere. There's a lot of programs. I'm sure anyone who's been in industry for more than a few years knows about programs that had to be backburnered because there's not resources. There was another program that was more important at the time. And we wanted to make sure that these things saw the light of day, got into companies where they could be taken forward. And once we had done we'd put together the last collaboration or asset sale, then that's when we figure we're out of business as Imago Pharmaceuticals. We had done pretty much what we hoped to do with Imago Pharmaceuticals. And that was when I started getting bored. And it also gave me the experience to know what works and what doesn't work in starting a company and what was different about what I needed to do for Nitrase Therapeutics than what we had done for Imago Pharmaceuticals. So that was when I started formulating the idea of starting NITROME and then Nitrase Therapeutics.
There are so many things to unpack in here. Let's start with the first one. How would a scientist like yourself who was working with Ilan start that conversation? Hey, I'm interested in this program that I want to take on. What are the dynamics like and how does one approach?
Yeah, I think that's a good question. For me, I didn't really know how, so I just talked to whoever I thought actually could make the decision. So in this particular case, it was the head of research and the head of legal. Ilan at the time was, I think, a 5,000 person company. So I didn't know the CEO to go ask him about it. I didn't know high enough level on the BD side to ask them about it. So what I did is I went to the highest level people that I knew, which in this case were the head of research and the head of legal, and asked them initially when I was still there and knew that the programs were going to be discontinued. And then when I had left, on every other month basis, emailing them, asking if they're now ready to out-license the programs. So I've never gotten my MBA. I've never done any formal training on the business side. I just knew that I wanted to make sure that these programs saw the light of day some way or another. And with decisions like this, it has to be people fairly high up that have any influence to be able to make those type of decisions. So I went as high up as I could.
One thing that stands out of this conversation is how persistent you were.
Probably one of my worst and best traits.
That's an important trait as an entrepreneur and more important than getting an MBA, I believe, because things like management you learn, but that persistence, that comes with you being a scientist, I believe. Well, sticking to that subject, like before we move on from Irene and Ilan, what are the lessons that you learned that you prepared you for Nitrase from Ilan and Imago days, if there are 2 or 3 lessons?
I think with the Ilan days, I was young and dumb and bold and everything else. So the Ilan days, what I would say is I learned how to do good science in a biotech setting. I also learned that maybe I didn't know everything and should sit back and listen every once in a while, or at least solicit input from people above and below, and to incorporate those things into my thought process. For Imago, I learned everything that I need. Well, I shouldn't say that I needed. I learned everything that I applied to NITROME from Imago. It was a completely different company, but in starting Imago, I'll start with the big things and go down to the little things. With Imago, we were definitive that we were going to keep it non-dilutive. It was 3 of us, 3 women, which was phenomenal. One of the best experiences working with people in my life, just because we all got along. We all agreed most of the time, which to have 3, even Diana was a scientist by training, so she understood the science. We had pretty much agreement across the board about decisions we would make, but we did decide that we would keep it non-dilutive. Diana and Karen had full-time jobs elsewhere at the same time that we're taking Imago forward. I was the only one that did not. We wanted the decisions to be ours, is probably the best way to put it, as opposed to as soon as you bring in money from outside, then the decision isn't entirely your own. And we wanted to have the flexibility of the decisions being ours, and we live or die by them, essentially. We kept the company entirely virtually because we could, and because it suited our situation in that we were in 3 different places. Having said that, with NITROME, it have to be a wet lab company. We started out with a proof of concept and had to actually get the science to prove out what I thought was the case. So I knew I needed wet lab benches. I knew that I had to have really high quality scientists that I would bring in. And we knew that we were going to need consistent money over a long period of time, and eventually that we would need a lot of money. And we didn't have the benefit of having programs that we could sell to do this. So I knew that we would have to bring in venture funding from the very beginning, or some type of funding, outside funding from the very beginning. I was still in Jackson Hole at that point in time. So I knew I would have to come back to San Francisco for lab space. And also not only for lab space, but I'd worked with really, really, really good scientists here. And I knew I had to have really high quality scientists to be able to solve this puzzle. Pretty much almost the opposite of Imago, not because we did anything wrong in Imago. It's just a very different situation. But then down to the little things, payroll's a nightmare, absolute nightmare. I started out with Imago. I actually paid payroll myself. And we were in 3 different, so federal, 3 different states, 2 cities that we had to pay taxes to, and then something in New York about the railway system. I was doing that by myself, and it's not like everything's online. So you fill everything out one place. Part of it's online, and another place is by paper, and keeping up with that was an absolute nightmare. So I learned even during the Mongo days to go to something like ADP, but even ADP is limited. So by the time I started NITROME, it was definitive. I was going to have a service that took care of everything. So we moved to TriNet as soon as I started up NITROME. So even down to little things, I was glad that we had started Imago before I started NITROME, because NITROME is the one that I really truly cared about. This was so unique that I'm glad that I learned a lot of things not to do on Imago's time and sort of set us up for NITROME.
This is a very common thread that a lot of entrepreneurs, you end up doing everything from cleaning to accounting to taxes. We are so resilient. I think people need to realize that and understand that once you get in there, you'll do whatever it takes. And it's fascinating that Karen and Diana stuck with you. And that is one of the themes for me. Like, I bring female entrepreneurs like yourself because there are so many wonderful female entrepreneurs who've done some amazing things. And I made it a point to bring female entrepreneurs to tell the stories. So thank you for coming onto the show. Let's loop back into Nitrase now. What is the composition of the founding story? Did Karen and Diana come join you here? What is that team like?
I would have loved to have them come join, but they still had their day jobs. And I think I had almost run Karen into the ground. She was CEO. I brought her into Imago to be CEO of Imago, and she did a great job at it. And I actually asked her when I started NITROME if she would come over to NITROME as CEO, and she refused. We're still best friends, but it was literally, she couldn't do it. She was still full-time at the SMA Foundation. So there's just no way she could take on a CEO role as a full-time job for NITROME as opposed to a part-time job at Imago. So she helped me. She invested in NITROME. We still talk and come up with ideas that might help out NITROME. In terms of actually bringing them into NITROME, wasn't able to do that. Would have loved to have. But what that meant then is that when I started looking for funding in the middle of 2017, I had to do it all on my own. And that's hard. That's really hard to do on your own. So I was still in Jackson Hole at that point in time. It's not exactly known as the Mecca of biotech. You may not be aware of that, but it's not the case. But as it turns out, there's a lot of people who've retired into Jackson Hole who are very accomplished. So I started attending, it was a foundation or a group called Silicon Coular. They're a group of people, nonprofit, who are trying to diversify the economy in Jackson Hole away from just tourism, basically. And they were really good because all I had to do was walk up to the guy who led Silicon Coular and ask him if he knew of any people in the Jackson Hole area that were from the biotech pharmaceutical industry. And within 5 minutes, he had emails for me for probably the majority of the people in the Jackson Hole area that were or are in the biotech biomedical, pharmaceutical industry. I started setting up lunches with them to pick their brains, see how I could do this, see if there were people in the Jackson Hole area that I could approach for funding for NITROME. The good thing about a small community is it's easier to actually find people like this, and people are more open to helping out and providing advice. So that was the good thing about being in a non-biotech mecca in trying to figure out how to get NITROME started and funded.
Interesting that you've spent your life in pharma and biotech space for a long time, and then you still decide to go with local folks in Jackson Hole for that initial seed money and advice.
I should probably say I looked everywhere. I already had a lot of contacts in the venture world, but this was just a concept. At the seed round, I probably contacted 40, 50 venture firms, a lot of interest, but none that would actually invest at that stage in the company. Having said that, several that I actually contacted at the seed round, I went back to them at the Series A, and at that point in time, they were interested. But at the seed round, it's just way too early for a venture fund to become involved. So that meant that I had to go to angel investment networks and to high net wealth and wherever I could to try to find the funding that would get us that proof of concept in place.
Where did the first dollar come from?
The first dollar actually came from an angel investment group out of the middle of Wyoming. They're focused in Casper, Wyoming. And it's a group of, I think there's around 21 people who've been successful in their own careers. And they put together a fund to diversify the economy in Casper and to help entrepreneurs in the Wyoming area. But these are our representative of the board was actually a stockbroker by training, but there were a couple of people from the high tech or who'd sold their companies. But a lot of ranchers, medical doctors, no biotech people in this angel investment network, but they were willing to listen to me. They were intrigued. What I found out afterwards, I was never quite sure why they invested in NITROME. What they told me afterwards is that they believed in me. They're farming people, or a lot of farming people, or a lot of hardworking, do things from scratch and be proud of what you've done. And I come from that kind of background as well. So they built a connection there. The medical doctors could evaluate to a certain extent what I had, but then they did their diligence. They went out to the universities and got professors to evaluate what I had. And so they did their diligence before deciding to become the lead investor. Even with that being the case, they were still a little hesitant. We're in the middle of Wyoming. I'm proposing starting a company in San Francisco. It's not central to their mandate of diversifying the economy in Wyoming. But it was actually when I applied for lab space at NBC Biolabs, and NBC has a really nice program where they work with pharmaceutical companies, and pharmas have what are called golden tickets. So before I had several groups who'd said they would invest in NITROME, but none that had actually signed on the dotted line yet to get the contract going. I applied for AbbVie's golden ticket and we won. And I think that was the thing that actually made it so that it sort of pushed them over the edge. It's like, okay, a company like AbbVie, which is a pharmaceutical company, they're interested enough in this concept that they've awarded the golden ticket to NITROME. Maybe we should advance as well. With Breakthrough 307 making the decision to advance, then that brought everyone else into the contract and signatures. So it was each piece coming together. I had to do everything. I had to look everywhere. I looked for any supplement. I looked for any external recognition of what we had to make anyone who was somewhat interested, hopefully push them over the edge. Or even with AbbVie, first thing was just any way I can get any type of funding possible, I'm going to try to get. But then the validation initially was secondary to that. In the long run has ended up being the primary driver of what's been helpful for NITROME and Nitrase Therapeutics.
That is fascinating. You have a bunch of non-biotech people interested in the concept. It's mostly because of you and your background. And they were waiting for a signal from the AbbVie golden ticket. What do you think convinced AbbVie?
I think what AbbVie and these pharmaceutical companies are looking for is the next interesting thing. And it was a lot of money for us, but it's not a lot of money for them. So what they're looking for is the interesting company that's very unique from what they're doing themselves, and that could be the next big thing. They're allowed to get an inside view on a lot of these tiny little companies really early on. They get to hear about the research, see how it's going, long before anyone else knows about NITROME or the other companies that apply. So I believe in each of the golden ticket competitions that we're involved in, there were anywhere from 30, 40 to 50 companies that would compete for these golden tickets. And they typically give out 1 or 2 golden tickets each year. So I think they're looking for the one that they think at some point in time they might want to invest in. And then at some point in time after that, they might want to do a collaboration with and eventually even acquire is the long-term goal. It allows them to keep track of what's going on with the companies, sort of have an insider view before anyone else does.
Wonderful. Let's go to the next stage. You got the golden ticket, you move back to San Francisco to start research. And what was that journey like in terms of, were there milestones that you're trying to hit in terms of those early stage angels to come in and invest money?
I'm a scientist by training, which means I'm not a marketing person. I'm not a salesperson. So I was actually very straightforward with my seed investors. Before they ever sign on the dotted line, I told them, we will identify this enzyme. I was pretty confident in that. I said, but we could identify the enzyme, and it could be something as soon as we identify it, we're like, no way would you ever want to inhibit this. So I was clear with them from the beginning that that's a possibility, that after a year and a half's time, their money's just going to go away. So they knew the risk. I should have been the salesperson and maybe bullshit a little bit more, but it's just not my. I thought about how to do that and I knew I couldn't do it. I'm a scientist through and through, so I couldn't do it.
It's hard as a scientist. It's hard.
It is.
Unless you're Elizabeth Holmes, yes. Well, she's not a scientist, I guess. She is not a scientist.
No, she's not.
I'll correct that.
No. So I made that clear with them before they ever invested. But then what they invested in was, we had very clear-cut milestones for the seed round. One was to identify the enzyme, synuclein nitrase. So the enzyme that catalyzes nitration of alpha-synuclein. I always figured if there was one enzyme, there was likely to be a class of enzymes. So once we'd identified synuclein nitrase, see if there is a class of nitrases. And then the third thing is to see if synuclein nitrase is druggable. Can you get small molecules to inhibit its activity? So those were very clear-cut milestones that we had at the seed round, and we accomplished each of them.
And this was an enzyme that was never before discovered?
Every gene in the human genome has been discovered. And so people think that with that being the case, that we know what each of those genes and translated proteins do. That's not the case. What was known about it is that a protein is translated. It had actually been crystallized. Its crystal structure was deposited in the public PDB. And there's so much information on the human genome and translated proteins that we're actually able to buy a lot of things that we needed to support this program as soon as we'd identified the enzyme. And part of that, the fact that its crystal structure was deposited in the PDB helped us identify homologs of synuclein nitrase that we thought could potentially act like synuclein nitrase. And then they became, our data internally showed that they truly were nitrases. And we're now up to the point where we have a whole class of enzymes, some of which we know are nitrases and others that we predict that we'll be able to show have nitrase activity at some point in time in the near future.
How long did it take for you to discover the nitrases?
The first one, the synuclein nitrase, it took 8 months. And it doesn't sound like much, but it was a grueling 8 months. There was only 3 of us in the company at that point in time, and it was a slog. I don't know if you've ever done conventional chromatography, but it's starting with large amount of material and going through trying to pick and choose, figure out what the best combination of each of the columns are, and at the very end, end up with a protein on a gel that you can identify by mass spec. And our enzymes are expressed at abysmally small levels. So all we know is it's greater than a 200,000-fold enrichment to be able to identify synuclein nitrase.
Wow. And who were these other 2 team members that joined you?
Yeah, so these are both people that I had worked with previously, actually at Hyperion. So Sami Hussein and Vu Deng. So Sami, I actually recruited into Hyperion. When we were at Hyperion, he was I had been told, and I found out that he had the best hands of anyone anyone I've talked to who knows Sammy has said he has the best hands of anyone in the industry. I found out that was the case at Hyperion, and I knew we needed someone who really had this type of ability to really do experiments exquisitely well. I knew I needed him at NITROME. So as soon as I made the decision to start fundraising, I started talking to Sammy. I told him what the concept was and trying to recruit him to get him to come into NITROME. I just knew I absolutely needed Sammy. So Sammy came in. He was second employee after me. So we brought him in to run the assays, set up really good assays to measure nucleotide nitration activity and to be able to track the enzyme activity throughout all of these thousands and thousands of fractions to get to a final endpoint. The second part of that is you need a protein purification person. I didn't have that person. Well, I did, and he wouldn't come over to a startup company that was on a concept. So then I had interviewed a person who wanted to come, who was a protein purification person, but I didn't know him. He was, right off of a biospace listing, which he probably would have been fine, but when I thought about it, do I want someone who I'm taking a risk with when I only have enough money to barely get through this? Or do I go with someone that I already know, I know what they're good at, and they have to learn protein purification, who's an electrophysiologist by training, but he's good at machines, he's good at tinkering, he's good at MacGyvering, whatever situation you're in. So I figured that he'd be able to pick up the protein purification. So we brought in Vu, and it was the two of them that do this whole process. And like I said, it was a painful process of identifying the first enzyme.
In the scientific community, a lot of people behave as if they've discovered everything, they know everything, and what every gene. I'm on the other side of this concept, and I think we humans haven't discovered a lot of unknown things. And
I agree.
And I was talking to Doug Crawford, and he agreed, and he was the one who recommended that I talk to you because of my thought process. And I'm glad there are scientists like you who are digging in deeper. So let's stick to the story again. 8 months, you're able to come up with the first class of nitrases. Is that when you raise your Series A?
That's when we started to raise a Series A. But even that process, it took, I think I started in June of whatever that was, 2019. We didn't get the final contract signed until April of 2020. We actually had our first investor was within a month or so of starting the process. Second one was within 2 months of starting the process. But then we wanted a really good consortium of investors. We wanted to make sure we have as much validation behind it as possible. We got our third investor, I want to say about 4 months after I'd started the process, but then to find who ended up being our lead investor, Sofinova Ventures out of France, it took longer to find that last group that would come in and co-lead. AbbVie Ventures was one of our leaders, and Sofinova took longer to find the right firm that was the right fit for us, and we were the right fit for them to lead the investment into the Series A. So even at the Series A level, it took whatever that was, 7-8 months to get funding in place. I think the economy, we knew something was changing from even when we started the Series A. It was just a feeling that there was a slowing down of interest in early stage biotech companies. And I think that was reflected in it. And then over the last month of putting the term sheet together and getting the contract in place, COVID set in. And so literally, as we were signing the term sheet, getting the contracts going, the economy was tanking, term sheets were being pulled. Even that process, there was nothing stress-free about it.
How big was the round, Series A?
It was originally a $38 million round. We'd always wanted it to be $45 million. We just felt like that's what would take us to IND for synuclein nitrase and to a development candidate for our second nitrase program. We only got to 38 as the economy was just about to tank. So we decided to finish it up at that point in time and then look at that final investor independently. If it came along, it came along. If not, then we wait for the Series B. But we were actually really fortunate, I guess about 4 or 5 months ago now in July, that we were able to bring in BMS as an additional investor, and that BMS brought the round up to $45 million.
Wow, amazing story. At Imago, you were completely non-dilutive. You have not raised any VC money at that point. And then here you are raising $45 million. It is pretty fast in so many ways to evolve as a founder in terms of raising that money. And did you have a process to select the investors that you want to bring in and those who you wanted to avoid? If you can walk us through the process, that'll be great.
For me, it was a lot of learning during that process. At the Series A, I knew that I wanted to have sort of that understanding that if we got this investor in, it was a substantiation of the technology of the nitrases. So I wanted it to not only be something where we were bringing in money, but that this shows that by this firm investing in us, that it's a validation of what we've done. And then also, up until then, I'd been running everything. I needed help. I needed a lot of help. So we wanted to bring in investors who could help me figure out how to run the company, what we should do with certain decisions, how do we diversify into the platform. And so we brought in investors and investment firms that we thought could help with this. Having said that, it's not like I was able to say, okay, I have 10 firms that I want to work with, and I pick these two. It was going after the right investors in the first place, and then pursuing those that were intrigued enough to be able to advance with us.
Very transparent enough when you're talking to the investors that I need help. And how can you help? Is that a question you posed, or
I did.
Were you scared of asking that question?
No, I'm a little older in this process, and I think part of that, I don't have the ego that maybe you think you have to have, or you do have as someone who's younger starting a company. The one thing I guess I could say to help people is you don't know everything. There's just no way. From what I saw, there was actually an appreciation that I knew what I knew and I knew what I didn't know. I think there was really an appreciation for that. In fact, I went through all the Series A discussions saying that I did not want to continue as CEO, that when the time was right, I didn't want to continue as CEO. And I think there was an appreciation for that, because often the people at the firms know better than the entrepreneur whether they're capable and should stay on as the CEO or not. So I think the fact that I preempted them on that, I think there was an appreciation for that. I think that it actually helps them to sort of evaluate, okay, this is what she is confident in and this is what she is not. I guess I'm past the stage of having to pretend like I know everything, because it's just not possible for anyone.
Egos are one thing, and the other problem is the fear. Most entrepreneurs want to appear that they're confident, they're capable of doing a lot of things. I think there's a balance they need to strike, and I don't know what that point is. I think you bring up good points. I think being upfront about it and saying, hey, I'm going to look for a CEO. That brings up an interesting question. Why did you not want to evolve into a CEO? What is the thought process behind wanting to bring a new CEO?
At the time, I was founder, CEO, CSO. I'd been doing everything from writing all the checks to getting the funding in to helping out in the lab, everything. And I knew I couldn't continue to do all of that for the long term. What I did do from the seed round through the beginning of the Series A round was bring in a really good team of scientists to make sure that we had as high quality of science being developed as we possibly could. And I consider myself a good scientist. I did all right as a CEO. I put all this together. I brought in a lot of money, but I'm not the best. I haven't done this for a lifetime. In my opinion, I think NITROME, I think it will be the next kinome, to be perfectly honest, just to be perfectly clear. I think it'll be just as important therapeutically as the kinome. So I've already built this really great scientific team. I think I can lead them well. Am I the best CEO, the best financing person, the best person to reach out and do the sales job of making sure that you take the company to the next level and get the next higher level of investment that we're going to need to really expand out the way we need to? I'm not. I'm just not. And I'm not interested in it, to be perfectly honest. I was split for so long, half the time having to think about anything financing and business related and the corporate attorneys and everything else. And the other time on the science. And the science, this is fun, even if it's hard. And this is like, and I had to think about it that much more because it's not natural to me. Whereas if we bring in someone like Pierre, he is a scientist by training, but he's been working on the BD and chief business officer, so business side of things, his whole career in the industry. So this is second nature to him. He's already shown he can do it. He's been good at it, brought in very large deals, been successful in helping with successful IPO. So this is something, in my mind, I wanted the business side to be just as good, just as phenomenal as the science side. And if I stayed in that role, and I have friends who've done that, where I have to be the CEO, I started everything, I have to stay there. And the company tanked because they weren't the right thing for that. So for me, it's more important that Nitrase Therapeutics succeeds in as big a way as it possibly can than that I stay in the front and center role where everyone sees me. It's just, that's not something I need to have. I would rather have the company be successful.
Wonderful. I think that clarity of mind and thought process is very, very important as a founder. As a founder, you got to do everything that you did. And I think knowing when to hand it off to somebody else. And how did you find the new CEO?
We actually went with the recruitment for this. We didn't want to bring in just anyone. It had to be someone really good. So we went with the best recruitment firm. We went with Russell Reynolds. And the team of women that we worked with at Russell Reynolds was just phenomenal. They did a lot of the legwork. They're connected to the whole industry, so they know who's even looking or interesting, or they interviewed us, myself and the board, to understand what we're looking for. And then they matched that with what they already know about people in the industry. And I'd say they brought us a couple handfuls of people that they thought were possibility. And every single one of them were like, yeah, I can see why you would say that. Yeah, it was actually really, it was expensive. I'm not saying it's a cheap process, but it was definitely worth it because we got the right person out of it.
Talk about the board. How big was the board at this point? And what was the composition like? How were they helping you?
The board is great. I like the fact that it was a lot of women, to be perfectly honest. It was nice. We have Henrietta Richter. She's the lead investor from Sofinova. We have Margarita Chavez from AbbVie Ventures. And then myself, now Pierre. I'm still an observer on the board, so I get to continue to monitor how that's going. And then we have John Bear from Dementia Discovery Fund, and then a couple of investors. So Katie Morgott was an observer.
Katie is on the board? Wow.
Yeah, she was an observer. But what I'll say is Katie is the reason all this came together. She was the person that I talked to constantly after we'd won the golden ticket.
Katie is awesome. I need to bring her here on the podcast.
Yeah, you do. You do. Yeah. Katie is the reason this is all in place, because she continued to follow up with me after the golden ticket. And she was the one that went through the whole process to get AbbVie Ventures on board for an investment into NITROME very early on. So Katie was instrumental in our development. No doubt about it. But in terms of who does what, the actual day in and day out work has to be done by people internally. We brought in a really good chairman of the board, Perry Carson. He was previously COO of Celgene. He was CEO of Pearl. He's been in venture. He just brought a wealth of knowledge for me from the corporate side that helped me out. I probably texted, emailed, called him at least once a day. He's probably really glad when Pierre came in, so he doesn't have to be bothered so much. So we have a really strong chairman who helped me out. I have scientists internally. I don't want to be bothering them in and day. I was like, what do you think we should do? What should we do with this equity investment? That's not the type of thing I want to bring to my scientists. So I talk to Perry constantly to get his advice. He manages the board. He's the one that's making sure that everyone's aligned and comfortable with how we're advancing. And then Henrietta is really as the lead investor, she's the one that people look to in terms of decisions about whether we're bringing in the right type of people and what should our equity look like, what should we do in terms of the next stages of development of the company. Margarita's from legal background, so her expertise usually comes in when there's something that we're concerned about from the corporate perspective, sometimes even into the patent portfolio side of things. And then John is our scientist. John Bear, everything that I think I could send him raw data and he would still be looking at that on a daily basis. So it's actually a really nice diversity of skill sets that are brought to help us that the internal employees take the company forward.
Wow, you have a phenomenal board. I'm glad you do have that team helping you.
Big difference.
Oh, yeah. Let's slightly shift gears. This is a long ride. How did you balance family and this gruesome startup journey? How did you receive?
Yeah, it's a good question. I would say I didn't. I don't have kids. We just have furry dogs. And I have a incredibly supportive husband. We've been married for a long time. Probably remember how long at some point in time. Since undergraduate days. He's like some of my investors. He's always believed in me. And I'm the one, he likes what he does, but he's not like, this is the thing I have to do day in and day out and think about all the time. So he's always been very good about all bad ideas off of him, see what he has to say. And so when it came time to start Nitro with Imago was fine. He loved it because we got to stay in Jackson Hole and he got to ski all the time. So Imago was easy. That was really a no-brainer for him. But when I brought up NITROME and that, this will likely take me back to San Francisco, and he's clearly not going to come back to San Francisco any more than he has to, he could have easily said, we're in this stage of our life, let's forget about it. But he didn't. He's been very supportive. Even when I'm now in San Francisco, the majority of the time, he takes care of everything that he can that takes any work off of my back. And he's proud of what I've done. He's supportive of what I've done. If we had kids, it would be a very different story. Honestly, I couldn't do it this way. He would have had to have moved back here and raised kids in one place or some type of compromise from that perspective. So not having the kids actually made a big difference, or being past the age where you have kids anyway. That made things a lot easier. But even with that being the case, during COVID, I barely went back to Jackson. It was one of few times in my life about any decision where I didn't know what to do. Do you fly in the middle of COVID? Do you drive 14 hours when I'm not that good of a long distance driver? For two people who'd never been apart since I started this company, we've been apart a vast majority of the time. So he had to be very supportive for this to continue to work.
Absolutely. This is something I tell most aspiring entrepreneurs that are trying to take that plunge, that they need that support in writing. You got to give them an idea of how difficult it is going to be, because it's hard. I have a 2-year-old and really, really hard. I'm trying to jump back into startup world, but I'm trying to gauge my wife's interest in supporting me.
Exactly. You don't have that, you're going to be in trouble.
Yeah. No, she's been very kind for my first journey, but I'm not taking my chance now. Thank you so much for sharing all those information. This is something I wanted to touch again about the naysayers. Even in the investor world, were there naysayers who didn't believe? What was holding them back? If you have to analyze hindsight, were the investors that were not willing to invest, and did the people say it won't work?
Let me just start sort of at a higher level. In terms of investors that didn't invest, I would say that I don't think there was a single investor that I went to that wasn't intrigued by the idea. How many times do they hear of a new class of enzymes that no one else has actually thought of? So I think everyone was intrigued. But I think in reality, venture is pretty conservative. I don't know how many times I've heard it's too early. And you hear about companies where you have a product on the market and it's still too early because you don't know how well it's going to do. So I think usually it was, for investors, it was more that we needed to get to a later stage. And even for a number at the Series A level, they still wanted us to get to the next step. A subset wanted us to get to the next step before they could invest. And I'm sure that will be true for another subset until we're IPO. I don't think I had a single investor who didn't believe there was a possibility here. They all most wondered how I could have come up with an idea that no one else had. But I personally don't really get the question, honestly. Everyone has ideas that no one else has at some point in time. It's just this one actually ended up being potentially really big. And like I said, have to ask the right question at the right time. But what I did have is a lot of people, it was more on the scientific side, really, that how could you have an idea that no one else has come up with? And since it was more of an ego thing, I would think, more than anything else. Since I didn't think of it, then you can't think of it type thing. And with people like that, I just figure it's not even worth the debate.
You don't respond to them. That's the best way.
I would present the data and show them what we have. And in my mind, what I think I'm actually very good at is developing science that's where we've thought of the potential issues before anyone else has. And if I show them solid data that in my mind proves what we have and they're still negative about it, then there's not much you can do about it, and it's not really worth the time.
Good point. One last closing question before my formal closing questions. This question is about the market. How did you look at the market size when you're pitching to these investors? Was that even something that you spent too much time and energy on? Or it's more of a technical risk at this point? Does a market come into picture? So what was your thought process on that?
It's a good point, and it was interesting because it depended on which investment firm I went to. At the seed round, so for the high net wealth, the angel investors, they all wanted the market comps. What's the potential out of this? Because they don't know. When I got to the biotech venture investors, I would say there was a subset of them that were almost offended that I was bringing the numbers up because they know the numbers as well or better than I do. And the thing that made it easy, I always had at least a slide on the therapeutic market for, say, synuclein therapeutic or Parkinson's disease, in case they wanted wanted to go through it. But at the biotech and venture level, there's so knowledge about these things. I think usually we would spend a tiny fraction of the time on that. Having said that, if you're proposing a therapeutic in a disease indication that no one has heard of, then you're definitely going to want to know what your market comps are.
Thank you for sharing that. Now to the traditional closing questions. I ask this to all my guests. If you have to get 2 lessons from all your experiences that will benefit aspiring entrepreneurs, what would they be? And if there are any mistakes that they should be avoiding, what would that be?
I think the first thing in terms of make sure you really want to do it. There was nothing easy about this process. The reason I did it was because I had to do it. Absolutely. There was no question I had to do this. If I went to my grave without doing this, it would have driven me crazy. So make sure you really want to do it. And then in terms of what I would have done differently, I got as much help as I could. And it may be different for someone else who's not starting at the proof of concept stage, but I would have gotten even more help if I could have. If I could have convinced someone to take on either a BD or a CEO type of role, I would have done it. I would have had more involvement because I would say there was 3 years where I worked night and day. On everything with NITROME. And it's exhausting. It's very exhausting. I tried a lot, but I would try even more to get even more help at the early stages to take more off of me. And also just so you have someone else to bat ideas off of. And then mistakes, I assume there's probably always mistakes. I don't know. We're where I wanted to be. So I can't think of mistakes at this point in time other than things that I would have, getting more help. And if I had not been able to do the things I wanted to do, like bringing in a really good team of people, then right now I would have a lot of regrets, but that wasn't the case.
Great. If you have to name 1 or 2 people that played a significant role in your startup journey, be both positive or negative, even negative ones help advances, who would that be?
Well, Sami Hussein, I needed him. He's been key to everything. He was our number 2 employee, and he was and still is key to everything. And then I would say because of how uncertain everything is at the beginning, chairman at the seed round, Ephraim Heller, he was in the Jackson Hole area. He was one of the people that I was introduced to. He's been CEO of a number of companies in the medical device space. So a little disconnected, but he's been through the process before. So I would initially bat ideas off of him and ask his advice. And after a short period of time, I asked him if he would come on as a chairman of the board. He had to think about it because he was just getting another company going, but he agreed to do so. And he sort of taught me what things I needed to do above and beyond what we had done at Imago. And he helped find some of the initial investors, some of the high net wealth investors that put a lot of money in at the seed round. So probably the 2 of them, just because early on is the really critical. Once you start to gain a little bit of momentum, then you have more people who are helping out. But early on, those few key people are make or break.
Absolutely. Thank you so much. You've been very generous with your time and sharing your lessons. I wish you and the team a lot of luck, and I hope lots of drugs come out of your company soon.
Thanks for having me. I really appreciate it.
Thank you for listening to this episode of the Lab to Startup podcast. You can find links to the resources and programs mentioned in these episodes, connect with Naresh, or subscribe to this show at labtostartup.com.